for the introduction. Dopamine has long been recognized as a key neurotransmitter in a wide range of cognitive control functions, including reward processing and decision making. It is well known that dopamine modulates the information flow within the reward circuitry consisting of the mesolimbic system and prefrontal brain regions. There's also increasing evidence that there are individual differences in dopaminergic activity that are thought to be affected by genetic variation in the dopaminergic pathway. So in the study, we investigated the biological mechanisms mediating individual differences in dopaminergic brain function. We created biologically informed multilocus genetic profiles representing the variability in dopamine signaling across multiple genes. The neural target of our study was variability in the responsiveness of the ventral striatum and the ventral tegmental area, which are key regions in the mesocorticolimbic system supporting reward-related behavior and motivation. The genetic target of our study was dopamine, which is released from neurons in the VTA and has a key role in modulating the responsiveness of the ventral striatum. So we included 6 genetic loci within our multilocus genetic profiles. All 6 genetic loci were carefully selected based on their previous associations with functional changes in dopamine transmission and ventral striatal reactivity, namely the DRD2 TAG1A, the C957T, the DARPP32, and the so-called SNP2, SNP17, and SNP19, and its labeling is according to a previously published paper from Zhang et al. In 2007. So in the study, 172 Caucasian healthy subjects participated. All subjects underwent fMRI while performing a reward-based decision-making task. Subjects had normal or corrected to normal vision and no history of neurological or psychiatric disorders. Human DNA was isolated from saliva samples obtained from all participants, and genome-wide SNP genotyping was conducted. Since animal and human studies provided evidence that dopaminergic neurotransmission is modulated by sex hormones, we were also interested in the effect and the interaction of gender differences on and dopamine profile scores on function, activity, and connectivity. So across all 6 genetic loci which went into the multilocus genetic profile scores, a score of 1 was assigned to relatively high dopamine genotypes, a score of 0.5 to intermediate genotypes, and a score of 0 to low dopamine genotypes. And finally, these scores at each locus for each participant were then summarized and to compute an individual profile score for each participant across all 6 genetic loci. And after that, subjects were divided into groups depending on the median split of the score, resulting in a group with low dopamine profile and a group with high dopamine profile. The group with low dopamine profile included 57 females and 27 males, and a high dopamine profile consisted of 54 females and 34 males. So our subjects underwent fMRI by performing a reward-based decision-making task, the desire-reason-dilemma paradigm. The experiment started with an operant conditioning task outside the scanner in which squares of different colors were conditioned as a reward. And the goal of this procedure was learning of response-reward contingencies for the next phase of the experiment. And in the end of the conditioning phase, subjects have learned that the red and the green stimulus were always rewarded. During fMRI scanning, subjects had to follow specific task requirements depending on 2 different types of blocks to successfully maximize their overall outcome at the end of each block. Overall, subjects had to collect all target stimuli, which were indicated by a Q at the beginning of each task block. And in addition, as you can see on the left side in the desire context, subjects were free to collect the previously conditioned reward stimuli for immediate bonus points, which were added to the additional points at the end of each block. In the reason context, as you can see on the right side, they were not allowed to collect the previously conditioned reward stimuli. They had only to collect the target stimuli, otherwise they would have lost all collected points. So the context changed after every second block and was indicated by a Q. And first, we could show that individual dopamine profiles and gender modulated the reward-related activation in the ventral striatum and VTA. We found significant main effects and an interaction effect in both brain regions. Furthermore, comparisons of gender revealed an increased reward-related activation in the ventral striatum and VTA in females with low dopamine profiles compared to males with low dopamine profiles in these situations when it was allowed to collect the previously conditioned reward stimuli. Furthermore, in situations when it was not allowed to collect the previously conditioned reward stimuli, females with low dopamine profiles and also females with high dopamine profiles showed a reduced suppression of reward-related activation in the ventral striatum and VTA compared to men. So in the second step, we extracted the mean parameter estimates of the ventral striatum and VTA and correlated them with individual dopamine profile scores. And this finding was really interesting and surprisingly. For the low dopamine profile group, we found a negative correlation between parameter estimates of the right VTA and dopamine profile scores in females. And for the high dopamine profile group, we found a positive correlation between dopamine activity, dopamine-related VTA activity, and dopamine profile scores in females. So in other words, we found an U-shaped relation between VTA activity and dopamine profile scores, but only in females. To further uncover the potential modulation of bottom-up activation in the VTA by prefrontal brain regions, we also conducted a psychophysiological interaction analysis using the VTA as seed region. And we found a stronger negative functional coupling between the VTA and the left anteroventral prefrontal cortex in females with high dopamine profiles compared to females with low dopamine profiles. And again, we extracted the mean parameter estimates of the functional coupling with the left anteroventral prefrontal cortex and correlated them with individual multilocus genetic scores. And we found a positive correlation between the functional coupling and the dopamine profile scores, and also again only in females. Meaning the higher the dopamine profile score in females, the stronger the negative functional anteroventral prefrontal functional coupling. So in conclusion, we could successfully show that the multilocus genetic composite approach is successful to show that individual multilocus genetic dopamine profiles modulate reward-related activation in our key regions, and also that there's an interaction between gender. More specifically, we found that females showed an increased activation and also a reduced suppression of reward-related activation depending on different task types. And furthermore, we also found an U-shaped relation between VTA activity and multilocus genetic profile scores in females, suggesting a compensatory mechanism depending on the dopamine turnover. And finally, we could also show that the functional connectivity between the VTA and the left anteroventral prefrontal cortex is modulated by dopamine profiles in females. So I was really fast. So in the end, I would like to thank my collaborators and my colleagues and all the people who contributed to the study. And I would also like to thank all participants who took part in the study. And I would like to thank you for your attention.